Hello, this is our lecture number 40, and this is on the biology of learning and memory, and this is a very exciting area of research, and lots of really recent work, as well as some older work that we'll be discussing, key experiments, and it's an area that has, very strong implications for understanding various behavior disorders that, for example, have to do with the loss of memory that may occur, for example, with advancing age in disorders like Alzheimer's disease, for example. So let's move along with this. We have lots to talk about, lots to get through, and again, a lot of interesting experiments. So let's begin this journey. This really is the new frontier, I think, in terms of trying to understand the biology of behavior, because there are techniques that have been developed in this area that are now being used very widely in many different areas of behavioral neuroscience. Some of this work, in fact, Nobel Prize winning work. So again, we're trying to understand the biological basis of the mechanisms that are involved in learning in memory. So some fundamental questions concerning this area. Certainly one question is, what are the biological factors that are involved in memory processing? Where, in what part of the brain does this take place? What kind of behavior disorders have helped us to better understand learning and memory, and there are a number that we could take a look at. And also, what kind of treatments are out there now based upon basic research by behavioral neuroscientists that have helped us to understand and hopefully treat those individuals who suffer from memory loss. So again, we'll work through these questions as we go along. So in this area of the study of learning and memory, behavioral neuroscientists believe that there's some kind of a physical trace for memory. Every time you engage in a new act of learning, there's something that's fundamentally changing in the brain. And some believe that it's a change in the way connections are taking place in terms of synaptic connections that are taking place in the brain. Some believe that the actual morphology of the brain is changing. Some believe that there's a change that occurs in neural activity of the brain, and some believe that the biochemistry of the brain is changing. But in all these different facets of research, again, by behavioral neuroscientists working in many different areas, there's very strong agreement that there is some kind of a physical change, but they don't have agreement in terms of which of those changes might be the essential ones. So let's work our way through some of the research that has been done in this area. So where does learning and memory take place? This is something that's been called the search for the Engram. Where does this learning take place in the brain? Where does it wind up in the brain? What are the actual physical connections that are taking place? The connections that are made between something that we actually do and some kind of a consequence that we have for that particular behavior. This is the essentials really of understanding things like classical conditioning and operant conditioning. So again, this has been called the search for the Engram, and certainly one of the very important historical figures in this area was a psychologist by the name of Karl Lashley, who many years ago engaged in this search for the Engram, and he did so in a couple of different ways. One way was that, and again, his work was done with lower animals, it was done with rats, he performed what he called cortical knife cuts. So what he would do is he would train animals in this very complicated maze, and then after they had learned this response, he would cut parts of their brain surgically. In the blue, what you see here are knife cuts that he makes. Again, one knife cut per animal. So one animal gets one here, another one gets one here, another one gets one here, another one here, here, and so on. You get the picture in terms of what's happening. And then what he did was to test these animals for their ability to remember what they had learned. He did it, several days after the animals had recovered. What he also did was to take other animals and perform what we call tissue ablations, in which he would actually remove parts of the brain. And again, he would do it, remove a small part of the brain in one animal, then another part of the brain in another animal, and so on. And he would perform these tissue ablations, after the animal had learned this response in this complicated maze, and after it recovered, he would test the animal to see how much they were able to remember. What Lashley finds at the time proved to be kind of shocking. What he finds is that it doesn't really matter what part of the brain he cuts. It doesn't really matter what part of the brain he takes out. The animals seem to remember very well what they have learned. So he, on the basis of this finding, he proposes the law of what he calls equipotentiality. And what he means by this is that all parts of the cortex are contributing equally to learning. One part can't really substitute for another part. What he also proposes is another law, which he calls mass action. He says the cortex works as a whole. Performance improves when more of the cortex is involved. Indeed, he finds that the more of the cortex that he takes away, certainly this is starts to have an effect in terms of producing deficiencies of memory. But for the most part, he believes that the cortex works as a whole, and indeed, certain parts can substitute for another part, but it's working as a whole. Lashley was wrong in some major ways. He assumed, for example, that this memory trace, the engram, was only occurring in the cortex, which we know now is not the case. And he also made a mistake in assuming that all memories are the same physiologically. And that too, as we will see, may not be the case. But this was pivotal work in this area at that time. As crude as it was still very important work in the field of behavioral neuroscience. Then comes along another scientist out at UCLA by the name of Richard Thompson. And he begins working with a very interesting model in which he explores classical conditioning and Eyeblink response in rabbits. So the way in which that works is this. You see a rabbit that is in this device that you see here, in which it's an automatic response for an animal to blink its eye, to temporarily close its eye when a puff of air comes towards its eye, as it does here. That's an automatic reflexive response. But then what Thompson is going to do is he's going to combine a sound that's going to come on, a tone that's going to come on shortly before the puff of air comes towards the eye. And if he does this enough times, what he finds is that eventually the tone itself, in the absence of the puff of air, produces classical conditioning. Again, this by definition is a classically conditioned response. So let's take a look at this a little bit further and explore a part of the brain that Thompson investigated. It's part of the cerebellum. It's this area that you see right here, which is called the lateral Pontine nucleus. Also just, the abbreviation LPN, lateral Pontine nucleus of the cerebellum. And what he finds is that during classical conditioning, there's a tremendous amount of activity that takes place here in the LPN when he measures it. If you take a look before conditioning, that is, you have the tone, followed by, here's our tone, presentation of our tone. Here's the puff of air. Here's our Eyeblink response that you see here. This is what happens before conditioning takes place. But what he finds, what Thompson finds is that after he pairs that tone with the puff of air a number of times, what he gets is the tone itself starts producing this starts producing the Eyeblink response before the puff of air even is administered. So again, this by definition then is a classically conditioned response. And then what Thompson does, in another very interesting experiment, and again, this is after conditioning takes place here. Again, you're seeing this tone being able to elicit the response, again, before the puff of air actually comes. Again, by definition, that's classical conditioning. So the lateral Pontine nucleus, lots of electrical activity occurring during learning of that response. And what Thompson does in another experiment is, after the animal has learned this response, what he does is he surgically removes the lateral Pontine nucleus. And what he finds is that the animal no longer exhibits the classically conditioned response. Again, here you have our tone. Here's the puff of air. The animal's not responding now to the tone. The classically conditioned response has been forgotten. So again, for a simple type of learning like this, classical conditioning, Thompson is able to find that it is located in the cerebellum, in this area that we call the lateral Pontine nucleus. So this was the first time that a scientist had been able to take a type of learning and memory and specifically identify what part of the brain was crucial for it. So again, the lateral Pontine nucleus, this is a major advance then at this particular time. Donald Hebb, another pioneering scientist, begins to explore what he calls two different memory systems, one a short-term system and one a long-term system. The short-term system, you'll also see this acronym STM. This lasts up to about 20 seconds. It requires a lot of rehearsal. You have to keep rehearsing it over and over and over again. The storage capacity of short-term memory is really, relatively limited to a specific number of items. Hebb says long-term memory, on the other hand, lasts 20 to 60 seconds or a lifetime. And it's anything that happened in the past which you can remember. So indeed, there appears to be two different memory systems, according to Donald Hebb, a short and a long-term system. Hebb believed in what we now refer to as a reverberating circuit. He believed that the shift that occurs from short-term to long-term memory and what he called consolidation of memories is due as a consequence of this circuit that you see here, in which neural input continuously is going over this pathway that we see here, okay, reverberating circuit. As that memory reverberates, again, around that circuit, it is causing a permanent change in terms of chemicals that are being produced, permanent change maybe in terms of the structural makeup of the nervous system. But this is, this is his theory as to what is actually happening. And he's really one of the first to begin to talk about this process that we call consolidation. That is, these short-term memories, if you rehearse them enough, eventually you're going to get into long-term memory, and they're going to be there forever. Indeed, later on, Hebb proposes that everything that we learn that ultimately gets into long-term memory, it's there, but we just have trouble sometimes accessing it, retrieving it. So again, Hebb also plays an important role in this story. There have been a number of important advances in memory research. Again, I want to list these for you, and then we're going to be talking about each one of them. Certainly the manufacturing of new proteins in the brain, that is thought to be important for memory processing. A phenomena called neural potentiation, thought to be very important, and certainly the research that's been done in what is called optogenetics is crucial for understanding this. State-dependent effects, we will talk about this. Morphological brain changes, some believe, there's structural changes that are taking place in the brain when we learn something new. And then there's been some more recent work exploring what we call working memory and the physiology of working memory. We'll take a look at that. There are syndromes that have helped us to understand memory, things like Korsakoff syndrome, for example, Alzheimer's disease, and certainly the research that's involved in trying to improve memory with acetylcholinesterase inhibitors, for example. So again, we'll take a look at all these things, as we go along in this research. So one researcher back in around 1980, between 1980 and 1985, Gary Lynch, begins to explore what happens in the hippocampus of the brain in response to electrical stimulation. So when he stimulates the hippocampus, one of the things that he finds is that he gets this dramatic growth changes here, what we call dendritic sprouting that you see, that you don't get in an animal whose hippocampus has not been stimulated. Now, as it turns out, the hippocampus is very important for memory processing, and he's really the first individual to suggest that one of the things that happens during memory is that you get this profound neural activation that is occurring of certain circuits, and it's producing growth changes in the brain in terms of this dendritic sprouting that you see here. There's also some really interesting work that has to do with the buildup of certain proteins in the brain that may in fact interfere with learning. For us to understand that, it's important to take a look at an experiment that was done, a classic experiment that was done some years ago by a researcher by the name of Keppel. And here's how this research went. Subjects were asked to learn some nonsense syllables and adjectives, like, for example, lum and happy. And then they did this in a masked way, that is a practice over a short period of time, or in a distributed way, in which they would practice, but then it would take place over a much longer period of time. So, for example, subjects studied this list of nonsense syllables and adjectives, and for some, they practiced it 8 times in the same day. That's what we call mass practice, when it's being done essentially all at once. And for others, it was just 2 times a day for 4 successive days. We call this distributed practice. And then what Keppel does is he takes a look at the ability of these subjects to remember what they had learned. For some, it was a 1-day delay. For some, it was a 1-week delay. Again, testing was 24 hours after the original learning or 1 week later. We'll take a look at what happens in the mass practice, as you see here. Memory 1 day later is pretty good, but memory 1 week later is substantially lower. That's in the masked condition. In the distributed condition, you see, once again, 1 day delay, pretty similar to what you see in the masked condition. But with a 1 week delay, these individuals engaged in this distributed practice are able to remember quite well, whereas in the mass practice situation, they are not. Now, this has an interesting parallel to what seems to be taking place biochemically. Repetitive learning tasks in lower animals activates a gene that produces a protein which is called protein phosphatase 1. And in mass practice, you get a huge buildup of this PP1. PP1 builds up when an animal repeatedly attempts to learn a task within a short period of time. Again, in our mass practice condition, PP1 interferes with the ability to retain what is being learned. However, if you take that same task and you spread it out over time in this what we call distributed practice, that chemical is allowed to decline. The PP1 declines between learning attempts, and the animal is able to learn better overall. So the message in a lot of this, in terms of learning and memory, practical implication, especially for college students, it's better to study over time than it is to cram for a test. So engaging in this distributed practice indeed seems to really promote learning. So that's an important takeaway message. And these findings of Keppel and then our assessment of protein phosphatase 1 is, I think, really interesting in terms of what is happening in terms of memory processing. Here's an important technique that you're going to be reading about as you go along, and it has to do with what's called Fos Immunocytochemistry. And Fos, F-O-S, is a marker. It's a marker that indicates that recent neural activity has taken place in the brain, and new proteins have been formed. If cells are actively engaged during a learning task, then this is going to be increased evidence. There's going to be increased evidence for elevations in these new proteins that are being produced in a lot of areas, but namely in the hippocampus, where we know, for example, that animals that are exposed to a novel stimulus compared with animals that are not, they show elevations in Fos activity. And take a look at it that you see here. This is an animal that's now been exposed to a novel stimulus, and you get these little dots here indicate a lot of Fos activity that is taking place. So you're getting these new proteins now in the hippocampus that are forming, and this is in an animal that's not exposed to that novel stimulus. So again, Fos activity, another way of assessing brain changes that may be involved in memory processing. And now we get to the Nobel Prize winning work of Eric Kandel, who was a real pioneer in this area, won the Nobel Prize in physiology or medicine in 2000. And he was really the first individual to establish a molecular basis for memory. And he wrote a book that I highly encourage you to take a look at. It's called In Search of Memory, which is really his journey in terms of how he was able to make these very important discoveries. So he works with the sea snail. Here's a sea snail that you see here. And they are really great to work with in this kind of research because their brain cells are very large. And they show these responses that are called conditioned defensive reflexes that are known to be serotonin dependent in the brain. You get withdrawal that takes place in aplesia of their siphon and their gill. Here's the siphon that you see here. Here's the gill. And again, when a part of the body is touched in the C-SNEL, those are going to withdraw. And what he does in this work is he's able to demonstrate habituation, dishabituation, and sensitization. These are three very basic kinds of learning that take place, and he's able to map out precisely what is happening in terms of these very large brain cells that are responsible for this type of learning to take place. So this was, exceptional work that was done at this time, which ultimately leads to him receiving the Nobel Prize for his work. So when you take a look at an element of the research that he conducted, he was recording single neurons in the hippocampus. And again, here we're recording from this cell body that you see right here when these excitatory synapses are being stimulated. And if you take a look at this figure that you see here, this shows current flow in the postsynaptic cell as a function of time. So when you present a very brief electrical stimulus to this cell that's coming from the hippocampus, and again, this is work that has been conducted with mice. Here you see a response that takes place as a consequence of that stimulus. So again, this downward deflection means that there's a change that's occurring in neural activity. What he does then is he waits an hour and stimulates these excitatory synapses again. And now look at the response. It's huge, right? And indeed, he believes, this increased sensitivity that occurs to the stimulation and what he calls long-term potentiation. He really believes, this is something that, even as much as several weeks later, he'll get this kind of response as opposed to this on the first time. And he believes that that's really the neural basis of memory, this long-term potentiation, LTP. So when you take a look at long-term potentiation, it's a protein-based morphological change that's taking place. Here's what a neuronal connection looks like before stimulation and the formation of new dendrites that occurs after the stimulation. So this is dendritic branching that is occurring in the axon, and again, he believes that this is the foundation for memory. The new field of optogenetics, and when I say new, I mean that it's probably about 10 years old now. There's two researchers that really co-discovered this, and Ed Boyden and Karl. And their research has been so important for this field, not just for the field of learning and memory research, but also the field of behavioral neuroscience in general. So optogenetics, how does optogenetics work? Well, here's how it works. If you take a look at algae that's in a body of water, for example, it's light sensitive. There's a protein in this algae that is very responsive to certain types of light. So it's an ion channel that's opening up in response to blue light. So when that blue light is detected, that channel is opening up. So here's what scientists do. They take the gene for this protein and they put it into the DNA of specific neurons in the brain. Individual neurons, okay? They're able to take this gene for this protein and insert it. Now, we know that neurons are communicating with one another by firing, okay? There's an electrical stimulus that occurs. Again, we talked about ion channels, and it's the opening and closing of those ion channels that's important for the action potential, the firing that takes place. Well, now with the insertion of that DNA from that algae, the protein from that algae, all we need to do now is to shine a light, a blue light, and it's going to cause those neurons to begin to fire. Here's an animal that has had an implantation of that DNA into specific neurons. Here now is the blue light that you see right here. Okay, so whenever that blue light comes on, it's going to activate that neuron, okay, that has that DNA that's been inserted into it. Though it does it very selectively, only in the neuron, again, where that DNA has been inserted. So with the right combination then of neurons being activated, you can activate a behavior, a very specific behavior, like movement, for example, or maybe sexual activity, or maybe the motivation to want to eat or to drink. And it becomes a fascinating way to be able to study the neural pathways that may be important for certain types of behaviors. So these two researchers, Zulio and Steve Ramirez, have done fascinating work where they've been able to turn on or turn off very specific memories. And I want to tell you about their research. It's a little bit detailed, but I think that once you get the basics of it, you'll understand it very well. So when we take a look on the left here of normal neural synaptic transmission, neurons become excited after sensing neurotransmitters in the synaptic area. And indeed, that is what is responsible for neural activation, normal synaptic transmission to occur. In the case of optogenetics, it's the light of a specific color that's triggering the opening of ion channels, okay? They're the same thing that is occurring with normal synaptic transmission, okay? So you're getting this neuronal excitation that is occurring. So again, now let's try to apply this to some very specific cases of learning. Engineering a memory with these LTPs, with what we call long-term potentiation and long-term depression. Take a look at the top portion of this slide. What we're going to do is we're going to inject channel rhodopsin into the auditory cortex, as you see right here, as well as the medial geniculate nucleus that you see here. And this area in the lateral amygdala is going to be the area where we are going to stimulate optically by way of this light, this blue light. Okay, so take a look here now. We have a rat that is trained to press a lever, and the rat will continue to press that lever in order to obtain food, for example, unless it is afraid to do so. So here's what we can do. Here's a rat pressing a bar in order to get food. But while it's pressing the bar, shortly after it presses it, it's going to get some mild shock to its feet. And it's also going to receive this optogenetic stimulation, okay, in the lateral amygdala area. Okay, here's the lateral amygdala area. So when the rat learns to associate now that foot shock with the optogenetic stimulation, right, it's not going to press the bar any longer. Again, it's associated those neural changes that are occurring as a consequence of the presence of the light, okay, with the foot shock that it received in the past. So it's not going to press the bar any longer, okay. So now, after a period of time, after now what we're going to do is we're going to produce long-term depression. And the way in which we do this is very weak pulses in terms of this electrical, this light stimulation that occurs in the lateral amygdala area. And what's going to happen is the animal's going to resume lever pressing. These slow, weak pulses are weakening that transmission of a signal. So now the animal starts pressing the bar again. But what we're going to do is now we're going to present fast, strong pulses, something that we call long-term potentiation, that's going to strengthen the synapse. And when we do that, the animal now remembers again fear of pressing the bar, right? So after the second round now of this long-term depression, the slow, weak pulses, right? Now the animal goes back and starts pressing again. So you've been able to turn on and turn off this memory very simply with fast, strong pulses that we call LTPs, and slow, weak pulses that we call LTDs. So again, this is fascinating work. Now we've identified very specific circuits that are involved in memory. Let's talk about another interesting area here, that of what is called state-dependent memory. These are memories that are associated with the physiological state when we're engaging in learning. And if you're in that same state later on, let's say several days later, and you're asked to remember what you learned on, several days prior to that, you're going to be able to do it well if you're in that same physiological state. So again, memory associated with a particular physiological state that you're in will be easier to recall, easier to retrieve when you are in that same physiological state. Let me give you example of what I'm talking about here. Let's say that you're learning, you're in a class in which they're teaching history, right? And you're reading material in a textbook, and you decide to have, let's say, a glass of wine, okay, while you're studying this material in history. You may recall what you've learned, let's say a week later. Okay, you're now being asked to remember what you recalled, remember what you learned, okay, from studying that history lesson. You're probably going to be able to remember it better if you have a drink, that is, you have some of that wine, shortly before the time you're asked to recall, okay, that material that you learned a week earlier. Because now you're in the same state that you were in when you learned, okay? And you're going to remember better than if you don't have that drink right before the time that you're asked to recall what you learned over here, okay? Now, I'm not advocating that you drink while you're studying. I'm just trying to illustrate the principle of what we call state-dependent learning. That, if you're in the same physiological state, okay, that you were in when you learned something, you're going to be able to remember that material better than if you're not in that same physiological state. Take a look at it this way. This is a summary, really, of some research that's been done with the hormone cortisol. Let's say that this is, you're engaging in some learning. okay? And let's say that you're in an aroused state while you're learning that material, and you're getting release of cortisol that's coming from the adrenal gland. That cortisol ultimately is going to go to a variety of different parts of your body, but namely it's going to go to brain. And one of the major receiving sites for cortisol is in the hippocampus of the brain. Now, let's say a week later, you're asked to remember what you've learned during this period of time. Now, if you're in a state in which you have a lot of cortisol that's being released, chances are you're going to be able to remember what you learned, and you're going to be able to remember it really well. But if you're in a state right here where your level of arousal really isn't very high, and you're not secreting very much cortisol at all, chances are your memory is going to be poor. There's lots of human and animal research that substantiates these state-dependent effects. And indeed, this has become a major important area of research in the field of the study of memory. Let's take a look at another aspect of memory that recently has been examined by an Australian psychologist by the name of Alan Baddeley. And he's been taking a look at working memory, and he believes that our working memory consists of these three important components, what he calls the phonological rehearsal loop, which really has to do with auditory and linguistic information, and what he calls the visuospatial sketchpad, and the central executive control system. And again, that's directing attention toward one or another stimulus, and that is, critically important for what we call working memory. Take a look at this very simple task that is used in order to explore working memory. It's called a delayed response task. It's a test for working memory, and it requires a subject to respond to a stimulus that maybe they heard or saw a short while earlier. So take a look at this. Take a look at this picture that you see right here. Here is a chimpanzee. He's taking a look at these two dishes that are in front of him. And in this dish, what we're going to do is we're going to place food. And then what's going to happen shortly after the chimpanzee sees the food being placed in the tray, both trays are going to be covered. And what we're going to do is we're going to lower this door right here for a short period of time, maybe several minutes. Then what we're going to do is to open that door, and we're going to take a look at whether or not the chimpanzee can correctly remember which of the food wells the food has been placed. And so the screen is raised, and the monkey now, typically they'll uncover the correct tray where they saw the food being hidden. Well, here's the important part of this research. If you take a look at the brain areas that are active during this task, tends to be the dorsolateral prefrontal cortical area of the brain. Again, here it is here it is here. So this is very active during this delay, okay? And it's believed that that's where working memory is taking place, that there's a lot of rehearsal going on in order to try to hold that memory in place. And indeed, it's in the dorsolateral prefrontal cortical area of the brain. So this is, some of the interesting research that's been done. In the next lecture after this, we're going to take a look at certain disorders that have really helped us to understand the biological basis of memory. So we'll take a look at that in our next lecture.